Hope everybody had a good break. Looks like we're not all totally ready to be back yet, but that's okay. We only got less than a month left, right? A couple weeks. Okay. We're gonna make it. So when we left off, we were talking about the stomach. So just to recap, now that we're talking about the GI system, we're talking about this hollow tube that goes all the way from our mouth out through our anus. So we're talking about all the food we eat, how we digest it, how we absorb it, the pathway it goes through, and all the various physiological processes that need to be involved here. So we talked about how we start with digestion in the mouth, we swallow, go through the esophagus, enter the stomach. We were starting to talk about some of the processes going on here in the stomach, particularly the production of this product called chyme, which is a mix of our food plus the gastric juices that the stomach is producing. So when we left off, we were looking at the gastric pits, which are these pits in the walls of the stomach leading to glands that are secreting a number of things, but particularly pepsinogen, hydrogen ions, which we're going to see in a little bit, are mainly coming in the form of like hydrochloric acid, and this thing called intrinsic factor, which is necessary for the absorption of vitamin B12. We also know that they're secreting mucus from the neck cells, so from the top of the pit, to protect the walls of the stomach from that acid that the stomach is secreting. And we also have some hormones secreted from the stomach, particularly gastrin, which is going to be partially controlling our digestive function. That's where we were through our travel through the GI tract. So next we're not leaving the stomach yet. We're going to talk about that environment. So we've swallowed our food, we've masticated it up, we've chewed it, we've swallowed it, and now it's in the stomach. So the stomach has this acidic environment. So it's a really low pH. The pH is about 2. That's why we need the mucus protecting the walls of the stomach. It has this low pH because we're secreting hydrogen ions into it, right? They're coming out of those gastric pits, specifically from our parietal cells. So they're secreting that acid, making the stomach acidic. This is important for a number of reasons. One is that one of our enzymes that is going to be breaking down proteins later on in the small intestine, called Pepsinogen in its inactive form, it needs an acidic environment to be activated. So we've thought before, especially when we're thinking about clotting proteins, about how we often have an inactive form of a molecule that needs to get activated in order to be useful, in order to do its job. So pepsinogen is going to be one of those. It's going to help us break down proteins for digestion. We need to have an acidic environment for that activation to happen. So that's part of what the acidic environment of the stomach is doing. It's also denaturing proteins. So basically denaturing, it means kind of unfolding them. You know, they're in this crumpled up, complex, three-dimensional shape. The acidic environment itself destabilizes some of the interactions between parts of the protein, so it starts to unfold. And it's also killing bacteria. This acidic environment is not hospitable to a number of things you might be swallowing accidentally. So that's a helpful sort of almost immune function here. So it's important that we have this gastric mucosal barrier produced by our neck cells, because the acidic environment of the stomach is important to your stomach's function, but we need to protect your actual tissue from that acidic environment. So we're going to have this protective layer of mucus. And in that mucus, it's going to be rich in bicarbonate, so kind of buffering out the pH along the walls of the stomach. So next we're going to move from the stomach into the small intestine. So we're passing through a structure called the pylorus. There's a sphincter here, slowly squeezing out little bits of the chyme, which again is your mixed up food, plus all those gastric juices, the acid, the enzymes, yeah, probably a little bit of the mucus, squeezing that into the duodenum, where it's going to continue into the other divisions, the jejunum and the ileum. Side note, when you're studying for, when you're studying for the exam, if it seems like something that you covered heavily in anatomy lab, that's probably not what we're going to focus on here in the lecture. So I know some of this stuff is doubling up. That's more for drawing linkages between what you've done in lab and here, rather than to explicitly duplicate things. Okay. So you've seen the small intestine physically before. Now, what you probably didn't talk about in lab is absorption in the small intestine. It's really highly efficient. So absorption is done basically in the first 20% of the small intestine. So it does not take your whole intestinal length to absorb things. We're going to have a really large surface area for absorption in the beginning of the small intestine, but also continuing throughout. So your small intestine, as you've seen, is a coiled hollow tube. You've probably maybe run it between your hands. If you have, you know, it's much longer than you might initially think. You've got about 8 to 10 feet of it in there. And it's between your stomach and ends at your large intestine, specifically at the cecum, before we move into the 3 parts of the colon. So the small intestine has 3 parts itself: the duodenum, the jejunum, and the ileum. I used to always flip jejunum and ileum, or be worried that I'd flip them. So the way I remember that is remembering the name of this corner with the cecum is the ileocecal junction. And that helps me remember that the ileum is the last part. I don't know if that's helpful for you, but it's helpful for me.
Is the omentum, that layer in front, that doesn't really make anything with the digestive part, more just the protection?
Yeah, the omentum is a sort of fatty layer, as you've seen. Yeah, it's a protective function, and it separates out sort of different parts of the abdomen. So if you had fluid escaping, right, leaking from your intestines, there are certain places it could travel, and other places it couldn't travel because of the sort of separation that creates.
Okay.
But yeah, it's not really involved in the digestive function. I think, I haven't really read the research on this, but I think that they have been finding that maybe parts of that are involved in hormonal production, or I think maybe I saw something about immune system, but I'm not totally sure. I know there's ongoing research in the field, but not my area. If you find anything cool out, let me know. Yeah. So we're in the small intestine, which is where the absorption is happening. Specifically, the absorption of nutrients is what we're going to have happening here. So when we say that we're increasing the surface area for this absorption to happen, part of that is about the length of the small intestine and all these coils, but part of it is about the cellular structure as well. So if we were to zoom in on the walls of the small intestine, we see that we have these villi, which are increasing the surface area. We can fit a lot more cells along these projections, right? We have more space. And there are also going to be blood vessels within this wall. And there's going to be something called a lacteal, which we'll talk about as we talk about fat absorption, but that's in this wall as well. If we look at the cellular level, those cells are going to have microvilli, which is going to increase the surface area of each cell as well. And so when we talk about the edge of these cells along the edges of the villi, we're going to refer to it as a brush border, right? It's a brush border because all these projections look like a brush. They have things brushed past them. So this is all for increasing surface area, because this is where we want all our food to come into contact with the walls so that it has a chance to be absorbed. Now, we are also going to have crypts here in the small intestine. So in the stomach, we had gastric pits producing enzymes. Here we're going to have crypts of Leverkuhn What they're going to be doing is secreting a fluid that's really rich in bicarbonate ions. And this is because, as we've just been talking about, stomach fluid is really acidic, right? We've made the chyme that's going into the small intestine quite acidic. Now, we don't want to have burning acid flowing through the rest of our GI tract, so we're partially going to be like neutralizing some of that acidity as we move into the small intestine. So the proximal parts of the small intestine, right? Proximal always means sort of nearer to the beginning. So those are the first parts of the small intestine that you're going to be going through. They're going to be secreting more bicarbonate-rich fluid, to neutralize that acid a little bit. We do want to keep that bicarbonate in our bodies, as we've been talking about our acid-base balance, right? We don't want to lose a ton of bicarbonate. We want to regulate that, keep that stable. So eventually that bicarbonate will be reabsorbed. So it's going to get absorbed later into the distal parts of the small intestine. So once we've neutralized that acid, we can kind of recoup some of our losses of bicarbonate. So as we think about secretions, a bunch of these secretions are going to be entering into the duodenum. So the duodenum is this first part of the small intestine. And this is where our pancreatic juices are going to come in. So we're going to take a look at the glands involved in digestion shortly. But the pancreas is lying posterior, so behind, and a little bit inferior to the stomach. So that's a large gland that we've talked about before. We talked about it in ANP1 when we were talking about hormones, specifically insulin and glucagon. Now that we're in the GI unit, we're going to be thinking about some of the enzymes that the pancreas secretes. The pancreatic juice is going to be coming from the pancreas, nestled back in here, entering into the duodenum. It's going to have a bunch of digestive enzymes in it to aid in digestion. And again, it's going to have bicarbonate. We're trying to neutralize that acidic chyme. Acidic because we put all those hydrogen ions, all that stomach acid into it. Yeah. Another thing that's going to be entering into the duodenum, this first part of the small intestine, is bile. So bile originally secreted from the liver. So we will also talk about the liver and its function as a gland. But we've thought very briefly about bile before when we thought about the processing of red blood cells. One of the times we mentioned bile before was when we talked about when we break down hemoglobin, we want to keep the iron, but we get rid of some of the other parts of the hemoglobin. So we talked about very briefly about bilirubin, which is going to be one of the things in the bile, not what we're going to focus about in this unit. But that bile from the liver is coming here into the duodenum as well. When we follow the path of bile, it's important to note that it's being secreted from the liver. And if you've had gallbladder surgery and removed your gallbladder, then it would just go directly from the liver into the duodenum. But usually, if you still have your gallbladder, it's going to get stored, produced in the liver, stored in the gallbladder, and then moved into the duodenum when it's needed. So we kind of have a reservoir of bile in the gallbladder. What we will be focusing on in this unit, so this is the main thing I want you to remember about bile, is that bile is going to contain bile salts. We're going to talk in detail about how these bile salts aid in fat digestion and help us break down big globules of fat, basically. So here's a picture of the liver. So one of the things the liver is doing is producing that bile and sending it to the duodenum. Another thing it's doing is basically, be careful about the word filter here. I don't mean it in the way that we used it with the kidneys, but blood coming from the intestines has to travel through the liver. And the liver is going to do some processing steps here. So when you were in lab, you probably looked at how arteries to the small intestine and the colon branch off the aorta, but all our veins are going into the hepatic portal vein and into the liver before they ultimately go through the hepatic vein and into the vena cava. And this means that all that blood that has been in contact with, in particular, the small intestine, which is what we're thinking about now, that's the blood that contains the nutrients we've absorbed from our food, right? That's going into venous blood. That's going back into your systemic circulation, going back to your heart with all those nutrients in it. But you may also have absorbed various toxins, things that you don't want in your body. And the liver is going to function to remove various things from the bloodstream, hopefully even before it gets to the rest of your systemic circuit. When we think about those nutrients too, we know that the liver can do things like store glucose as glycogen. So that's another example of the kind of processing step we're going to have going on in the liver. So don't worry about rememorizing all the branches of the vasculature, but do know that those nutrients and whatever we're absorbing from our GI tract are going into the veins, which are ultimately part of this portal system. So they're going into the portal vein and into the liver. Okay, so this is where this processing is happening. So continuing with the GI tract, after we've gone through our small intestine, so our duodenum, our jejunum, and our ileum, we're going to enter the large intestine at the cecum. Okay. So the cecum is this sort of bottom right corner where you would also find the appendix. So if you're worried you have appendicitis, right, it's often a sharp pain right there where your cecum is, because the appendix is an appendage off the cecum. Okay. So we have a sphincter here as well. Our ileocecal sphincter is controlling the flow of material from the ileum into the colon. A feature of the large intestine that can help us distinguish it from the small intestine, if we're looking at it, and it'll also have some sort of functional effects when we talk about movement of our products through the GI system, that we have bands of longitudinal, long, smooth muscle called the taeniae coli. It's bands of smooth muscle. So like the small intestine, the colon is a hollow tube. It's a little larger in diameter. That's why it's our large intestine. That's not about length, it's about how wide it is. And we name the parts of it after the cecum just based on what direction they're going. So we have ascending, going up, transverse, going transversely across, descending, going down. Then your sigmoid colon is a kind of S shape going into your rectum and then into your anus. Now, the purpose of the colon, at this point we've really absorbed our nutrients, right? So most of that was happening in the first 20% of the small intestine. But we've still got a long way to go before waste is excreted as feces. One of the things that's going to be happening here is really concentration of that mass. So concentration of feces into something that's, you know, relatively solid, preferably, right? So there's going to be some water absorption going on here in the colon to make that happen because the chyme is quite liquidy. So that fluid that was going into your small intestine, made up of gastric juices and food that you chewed up and digested, that's quite liquidy. We don't want it to come out all liquidy, so we're going to absorb a lot of that water, most of it. The colon also stores feces until defecation. So it can store quite a lot of feces. I think there's usually like a normal amount is like up to like 7 pounds of feces stored in the colon. Right. Okay. The colon empties into the rectum, and then the feces is going to be excreted through the anus. So this is another place where we're going to have these pairs of sphincters, just like we saw in the bladder, just like we saw in the esophagus. So we're going to have a sphincter made of smooth muscle, and we're going to have a sphincter made of skeletal muscle. So one we can control actively and one we can't. So the internal anal sphincter is the one you can't control. The internal one is made of smooth muscle. So that's a process under autonomic control, right? And the external anal sphincter is made up of skeletal muscle. So that's when you can control, you can actively prevent defecation through the external anal sphincter control. Now, in order to defecate, in order to excrete feces, both sphincters would have to be open, right? One of those sphincters is closed, like a closed door. You have to open both of these double doors. So now we're going to move on to the glands. I've tried to kind of mention them as we go through the regions you'd find them in, because I think linking up these topics together is the best way to understand it. But we're going to look at their insides and their products right now. So we're going to be secreting products of our glands into the lumen of the GI tract generally. So different parts of this system that we've just gone through are going to have different types of secretions going into them, but the overall kind of purpose of these glands is to produce enzymes that are going to help us chemically digest and then absorb what we're eating. So there are 4 sort of types of glands. I don't know if I'd really, I wouldn't memorize that number, I guess is what I'm saying. But we're going to start out in our mouth and in our face with salivary glands that are secreting saliva, the spit. We're going to have the pancreas. So the pancreas sitting behind your stomach is secreting pancreatic juice. So we know that's going into the duodenum. Those pancreatic enzymes are doing some digestion. And the reason I'm hesitant to really agree with this number 4 is that a lot of these fluids are coming from structures called acini, but this is more of a sort of general structure type that you might find in a salivary gland or in the pancreas, rather than an organ of its own. So the acini are these little sort of blind ends where we have clusters of epithelial cells that are going to be secreting these enzymes, basically. So this is just a generalized structure of a gland. So that glandular tissue, touch it if you find it kind of dense, but it feels or looks kind of like fat if you're looking at it. So that's our body of the gland. And inside, I'm going to have some type of duct ending in these cul-de-sacs, these little dead ends, where we have our secretory cells in the acini. So that is true of the salivary glands and the pancreas, but the liver has kind of its own thing going on. So we wouldn't call the structures we see in the liver acini. Starting with the saliva, like many of the fluids we're talking about, the saliva is rich in bicarbonate ions. So this is to neutralize anything super acidic you might eat. The saliva has mucus to lubricate food, make it go smoothly down your esophagus, and it also has enzymes in it. So 2 of the enzymes that are present in saliva are salivary amylase. This would be, I think, the more important one to remember. So the salivary amylase is going to start down by breaking up carbohydrates. So we know it's enzyme, and we also have some lysozyme here. This is basically just breaking down other things that you're eating. So remember the salivary amylase particularly. Next, we're going to the pancreas. So the pancreas has both exocrine and endocrine function. So exocrine meaning that we're producing enzymes that are going into the GI tract in this case. So sort of nearby function. Endocrine meaning we have kind of hormones going into the blood. So previously we focused on its endocrine portion in the islets of Langerhans and its production of insulin and glucagon. But now we're thinking about the rest of the mass of the pancreas. Those islets of Langerhans are just little islands scattered in the pancreas. So the exocrine pancreas is what's producing our pancreatic juice. Okay, so pancreatic juice is going to be produced in those acini, in those specialized enzyme or specialized epithelial cells that are secreting enzymes. Then they're going to go into the duct, which has duct cells lining it. Then we go into the named pancreatic duct. And then we're going to ultimately dump into the duodenum through a sphincter that controls ideally both the pancreatic duct as well as the common bile duct coming from the liver and the gallbladder. So the fluid that we're producing in this exocrine pancreas, like the saliva, is going to be rich in bicarbonate. Right? So now we're dumping into the duodenum, and we then have chyme, which is acidic, coming from the stomach into the duodenum. So we're trying to neutralize some of that kind of as fast as possible. Okay. We're also going to have a lot of enzymes present, specifically pancreatic amylase, which is going to continue carbohydrate digestion, lipases, which are going to start fat digestion, as well as some proteases and nucleases involved in protein and nucleic acid digestion. A fun fact about the pancreas is that relative to its weight, relative to basically how big it is, it's secreting more protein than any other tissue in the body. So it's really busy making these enzymes. It's making a lot of enzymes. And we'll talk about the function of each of those enzymes when we talk about the digestion of each of our specific macromolecule types. So this is not the only chance you get to see the names of these enzymes. So last we have the liver. So the liver has a lot of different functions. And in this unit, one of the ones we're focusing on is its secretion of bile, which is going to help us break down fats. But other things we have talked about a little bit are some of its processing of nutrients. We also talked about its processing of red blood cells. But as a reminder, because this is now linked up to the food we're eating, right? So as you're eating food, right, you're breaking down that food into its constituent parts, so into the macromolecules, and then ultimately into monosaccharides, amino acids, nucleic acids, and little bits of lipids, so triglycerides, cholesterol. Okay. Now, one of the things that the liver can do is store those products. So we can make glycogen from glucose. So the glucose that we absorb, we can store it for later. So we link together a bunch of glucose molecules. We create glycogen in the liver, which can then give us really fast glucose stores later on when we're ready to use that glucose. Amino acids can actually be converted into fatty acids by the liver, again for storage. Your liver can also make triglycerides and cholesterol as we are absorbing things. And it makes lipoproteins, kind of partially from those triglycerides. So a lipoprotein is just a lipid and protein kind of together. So it's processing nutrients, which are coming in through those veins from the portal system, right? So we absorb them somewhere else in the small intestine, and then they're coming into the liver through the portal vein, where it's processing them and probably storing them. But it has lots of other functions as well. So we also talked before about how it's removing old red blood cells. So it's pulling out the iron so that we can keep the iron for making more hemoglobin. But to do that, it has to break down the hemoglobin, has to catabolize it. And its byproduct of pulling that iron out of the heme group is it's generating bilirubin, largely from the globin part of the hemoglobin, which we refer to technically as a bile pigment. So that bilirubin gets eliminated in your feces. It's actually green in color when you produce it. So you've probably seen this in lab. It's kind of bright green, stains the gallbladder and your bile ducts green. That kind of turns brown as it moves through your GI tract. So that's what gives feces their color. The liver is helping you in general eliminate waste and toxins. So different medications, different drugs, all sorts of byproducts are going to hopefully maybe be filtered out by the liver. It's going to at least try. So we have various enzymes that can neutralize things in the liver. Also synthesizing various plasma proteins. So it's synthesizing albumin. It's synthesizing those clotting proteins that we talked about when we talked about the cardiovascular system. So sending various plasma proteins into circulation to do jobs throughout the body. It's also secreting and modifying various hormones. One of those is related to growth. So insulin-like growth factor is one of the things that's coming from here. So it has really a lot of jobs. The liver is pretty important. What we're really focused on for this unit is the bile part, because we're thinking about digestion. So the bile is going to help us digest fats. So you need to remember that the liver is synthesizing that bile and it's flowing out through our hepatic ducts, through the common hepatic duct, maybe getting stored in our gallbladder so long as we haven't had it removed. So cystic duct could send it into the gallbladder and then potentially back out. Ultimately, we're going to go through the common bile duct, join with the pancreatic duct, create this little sort of swelling called the ampulla of Vater, then go out through a sphincter called the sphincter of Bode, which is regulating flow of these juices into the duodenum. Okay. So summary, liver is producing the bile, gallbladder is storing the bile, bile ducts join up with the pancreatic duct, and the release of bile into the duodenum is controlled. Okay. If we look into the liver, we can think a little bit about how we make that bile. So the bile are the materials to make it are going from that venous blood coming through our portal vein. So we're picking up materials. They're going into the edges of these little substructures of the liver called lobules. So you can see along the corners of each lobule, we have a vein, we have an artery, and we have a bile duct. So we're bringing our nutrients in on those veins. The blood is going to pool in these structures called sinusoids. I have not been able to find a picture of them, but they're basically just pools of blood where we can have exchange of materials. So the cells that are going to be secreting and producing that bile are called hepatocytes. So hepato for liver, so basically just liver cells, right? They're going to secrete the bile into the bile canaliculi, so the little tubes that are going to be on the opposite side from the sinusoids. So the sinusoids are where we have like the blood pooling. We take the things we need to make the bile out of the sinusoids. We make the bile in the hepatocyte and then put it into the bile canaliculi, which are then going to drain into the bile ducts, and then they're going to drain ultimately into the common hepatic duct. So what you want to remember from here, really mainly that the hepatocytes are the ones that are making that bile from the products that we're getting into the blood, and then we're draining it into our bile duct system, our biliary system. So here's an overview of all the parts of the gastrointestinal system and what they are doing functionally. We're going to step through next by molecule type. So I think these were numbered wrong in your folder, but what we're going to move into next is the absorption packet. I know some of you guys take notes on paper, so since I think they were misordered in the folder but not in the schedule, does anybody need a printed out packet? No, we're good. Second to get to this absorption. We've moved through the GI system in order of its anatomical structure. Now we're going to pick out specific macromolecules and think about how they get processed individually as they're moving through the GI system. So I've been trying to give you sort of teasers as we were talking about the anatomy part so that you have something to build off of as we think about the specific enzymes now that we're thinking about specific types of macromolecules. So we're trying to digest and absorb a bunch of nutrients and also water. So mainly nutrients when we're thinking about the small intestine and mainly water when we're thinking about the colon. But in more detail, we want to think about how we're absorbing carbohydrates. We want to think about how we're absorbing proteins. We want to think about how we're absorbing lipids. Then we want to know that we're also absorbing vitamins, minerals, and water. All right, so here's where we are originally absorbing water, by the way. And when we talked about kidneys, we talked about reabsorption. This is the original absorption from what you're eating and drinking. This part, the colon, is absorption. Our kidneys were reabsorbing because of this first absorption step. So we're going to start with the carbohydrates. So the cells in your GI tract can actually only absorb monosaccharides. Right, so only monosaccharides, only these single units are going to get absorbed into cells. But most of the things you eat are not monosaccharides. So you're not really eating just spoonfuls of glucose and that's it for your carbohydrate intake. You're going to be eating a lot of disaccharides. You're going to be eating a lot of polysaccharides. And you need to digest those down to their monomer parts, right? To their monosaccharides, their smallest part, in order to get them into your body. So like physically into your cells. So to do that, we have to digest the molecules, which really just means break them apart. So this is starting in the mouth. So in the saliva, we have salivary amylase, right, which starts breaking down our carbohydrates. But you don't just keep carbohydrates in your mouth until they are completely dissolved. Swallow, this process is somewhat fast, so your carbohydrates are not fully dissolved in the mouth, which means that you have partially digested, partially dissolved carbohydrates traveling down through your esophagus, traveling into your stomach, traveling into your small intestine, which is where we have the next enzyme that's going to help with carbohydrate digestion. So in the small intestine, specifically in the duodenum, we're getting all that pancreatic juice coming through the sphincter of Oddi, right? So that controlled release of the pancreatic juice, all those enzymes produced in the pancreas going through the pancreatic duct, getting released into the small intestine, into that first most proximal part, the duodenum. And they're going to continue this digestion of carbohydrates. But a funny thing about carbohydrates is that digesting them isn't as hard as it's going to be when we're digesting lipids, when we're digesting fats. But our amylases are only going to be able to do so much as they break stuff down. So what they're going to struggle with is they can break between pieces of the chains, but they have a lot of difficulty. Basically, they can't break at the branching points. They struggle with these branching points. So as we start this digestion, we're breaking off pieces of our larger carbohydrate, but a lot of our storage molecules, particularly starch, so that's our plant form of storing glucose monomers, have these branching points, so we need to figure out how to break those apart as well. So the structures that have those branching points left on them are called limit dextrins. I think about this, I'm not sure if this is why they're called that, but you know, we have limited ability to digest. So I remember this limit dextrin term. Then we're ultimately going to have some more enzymes that we're not going to worry about naming that are embedded in the walls of the small intestine that are able to kind of break down some of these branching points as well. Okay. So our two enzymes produced by glands that are helping are the salivary amylase in the mouth, the pancreatic amylase produced by the pancreas, and then used in the duodenum and the rest of the small intestine. But they can only break the carbohydrates down so far. They have some trouble. So there are going to be other sort of accessory enzymes embedded in the walls of our small intestine that allow us to get down to that monosaccharide structure and ultimately absorb. Okay. So next we're going to move to the proteins. So the proteins are going to be broken down by proteases. So that's the general name for an enzyme that breaks down a protein. We can break down our proteases into a couple of different types. Okay. So our endopeptidases, they can break like interior peptide bonds. So we have amino acid, amino acid, amino acid, right? So they're going to work kind of like those amylases did. So they can break interior bonds. But what they're not going to be able to do is break off the amino acids at the very ends of our polypeptide. So that's going to be the role of our exo peptidases. Exo peptidases are going to be able to peel off amino acids from one end or the other end of a polypeptide. So we need both in order to get down to a single amino acid, right? Because if you think about breaking this chain, right? So if our first step is an endopeptidase here at 1, that would leave us with these 2 amino acids to each other. And that means that both of these are kind of an end. So we can't have another endopeptidase work here. This is where our exopeptidase, step 2, would need to break off one end from the other. So we're going to see these two types of proteases working in concert, working together in order to break up our polypeptides. The other thing that we're going to need to be aware of, we kind of think of as a class of protease, are our Zymogens. But they're a little different than the endopeptidases and the exopeptidases. What a Zymogen is an inactive form of a protease. So just like we needed to activate clotting factors, we need to activate our endopeptidases and our exopeptidases. So the Zymogen is going to be a general name for all of our inactive forms of proteases. So these inactive forms are stored in Zymogen granules, which get secreted from the cells by exocytosis. And so we can think of these as kind of like vesicles that secrete them by exocytosis. And then they're going to need to get activated. In most cases, we basically activate them by breaking them down. So that's what proteolysis is. Break them down a little bit, and that is what turns them into an active endo or exopeptidase. We're going to think a couple of specific examples, right? So you eat a protein, right? So you chew it up, so you break it down mechanically. We don't have any proteases in our saliva to start the chemical digestion yet. So you swallow it, goes through your esophagus, goes into your stomach, and now we can start to chemically digest that protein. So in specific, we have one enzyme in the stomach that is going to be digesting our protein that we've eaten, and that enzyme is pepsin. So pepsin is our stomach protease. Okay. So pepsin is originally secreted by the chief cells in the gastric pits. So these are our chief cells. They're secreting pepsinogen. So this would be like an example of a Zymogen, right? An inactive form of one of our enzymes that are breaking up proteins. So this would be a Zymogen. Example. So inogen, whenever we see that ending, that's a clue to us that we have some inactive protein, right? We saw that ending when we think about clotting, right? We saw plasminogen. We've seen this sort of ending before. Okay. So we need to activate that pepsinogen into pepsin. So the way we do that is by contact with acid. Now the acid is coming from the parietal cells. We talked about them in the gastric pits as secreting hydrogen ions, which they are, but those hydrogen ions are largely coming in the form of hydrochloric acid. So that's our stomach acid. So that hydrochloric acid breaks up or reshapes the pepsinogen enough that it activates it into pepsin. Now the pepsin can then get to work and start to break up our protein into smaller peptide fragments. The one cool thing that's also happening in the stomach at the same time, right? We mentioned that acid also denatures things, right? So the protein is kind of unfolding at the same time, and then pepsin can get in there and start to break it up. Pepsin here would be breaking it into smaller peptide fragments, so we might call it an endopeptidase, right? We're not just breaking one off the end here. Now, in the small intestine, we're going to continue protein digestion, but we're going to have some new proteases that we're going to talk about. So we're going to have trypsin, chymotrypsin, carboxypeptidase coming from the pancreas. And then we're also going to have some proteases embedded in the brush border, right? So we have some proteins that are stuck to the walls that are acting as enzymes. And we'll talk about these specific proteases and how they work and how they interact when we next talk on Friday.